make her ready.
Yeah, tell us.
Sir, what's will be conversation posted in shared to each other?
Okay. Hello. Sir. Any difficulty, sir? Oh. Okay, sir. Yeah, I'll ask. Yeah, yeah. Thank you, sir.
Welcome all of you to the information webinar on cutting-edge strategies in drug discovery, organized by Department of Pharmaceutical Chemistry, 7 Hills College of Pharmacy, Tirupati. First of all, I would like to call upon our beloved principal, Dr. M. Niranjan Babu sir, to address our today's international webinar. Hello, team.
7 years college of pharmacy. Dr. Niranjan Babu. Hello. Audibility is not available, sir. Good morning, 7 years college of pharmacy. Am I audible?
Good morning, sir. Yes, sir, you are audible. Your voice is audible, sir.
తెలీదు.
Hello. Hello. Is anyone there? Can you hear me?
Yes, sir. Yes, sir. Your voice is audible. You can hear me. Hello. Srinivas sir, is it audible?
అయితే లేనే లేరు అసలు మనకి.
Hello.
Good morning, sir. I'm listening. Wait for another few minutes.
Okay, cool. I've been in
Yeah.
confusion.
Yeah, some
of-
technical issues are there. We are going to
Okay.
sort it out, and shortly we are going to welcome you.
Oh, cool. No problem. Thank you.
సార్ నమస్తే సార్ నిరంజన్ బాబు సార్
Hello.
సార్ నమస్తే
Namaste. Good morning, sir.
వెరీ గుడ్ మార్నింగ్ సార్
Yeah, yeah. Now I think it is okay. Yeah.
Morning call. Sorry for the delay because of the technical issues. Start the webinar. So once again, I welcome all the delegates to our today's webinar on cutting-edge strategies in drug discovery, organized by Department of Pharmaceutical Chemistry, Seven Hills College of Pharmacy, Chemistry. Now, I welcome our Honorable Principal, Dr. M. Niranjan Babu, sir, Town Health College of Pharmacy, Tirupati, to address about the international regulation. Over to you, sir.
Yeah, thank you, Mr. Nagendra sir. Thank you very much. Om Namo Venkateshaya. Namaste. Good morning and welcome to our webinar. Today's topic, cutting edge strategies in drug discovery. I myself, Dr. M. Niranjan Babu, Principal, Seven Hills College of Pharmacy, Autonomous Tirupati, Andhra Pradesh, India. On behalf of Seven Hills College of Pharmacy, I welcome our today's chief guest, respected, honorable, humble and dynamic Principal of Sarada Vilas College of Pharmacy, Mysore, Karnataka, Dr. Hanumantachar Joshi sir. You are welcome, sir.
Thank you, sir. Dr. Niranjan Babu, sir. Thank you.
Thank you. And today's resource person, respected Dr. Srinivas Kantam, Senior Scientist, Measentech, Vancouver, Canada. Sir, you are welcome, sir. A warm
Thank you,
welcome
sir.
to you, sir.
Thank you. Thank you. Thanks for having me.
My dear respected faculties, registered delegates, all are welcome. We are excited to have you here and we hope you will find this webinar informative and helpful. I am also a big believer in power of webinar to connect people and share the knowledge. That's why I'm also so excited to be here today with you. Thank you everyone present here. Have a nice day. Jai Hind. Over to Nagendra sir.
Thank you,
Further
sir. Thank you for
proceedings.
your valuable introduction and inviting our chief guest as well as our speaker. Thank you, sir. Thank you. It's my immense pleasure to introduce and invite our chief guest, Dr. K. Joshi Hanumantachar. With all due respect, I cordially invite our today's chief guest, Dr. K. Joshi Hanumantachar, Principal, Sarada Vilas College of Pharmacy, Mysuru, Karnataka. Sir has completed his doctorate in pharmaceutical sciences from Guru Jambeshwar University of Science and Technology, Research, Haryana. Sir is having a valuable experience, more than 20 years in teaching and research. His research interests include neurosciences, behavioral psychopharmacology, herbal psychotropics, standardization of phytopharmaceuticals, Alzheimer's, and Parkinson's disease. Not only that, sir is having various number of research and review papers published, around 227 in national and international journals and conferences, having HNS 21 and INDEX 21. Sir is also having a total of 11 national and international patents and 7 design patents on the of UK, Australia, and India, and so on. Not only that, sir is also acting as editorial member for 11 international and national journals. Sir is also acting as a reviewer for 90 international and 32 national journals, and sir is also acting as the president, Alzheimer's and Related Disorders Society of India, Mysore chapter. Not only that, sir is also having numerous jewels of awards on his name to present you. I'm going to present the important awards that sir has had. Not only limited to this, there are numerous number of awards on the page of our today's speakers. The list goes like this: Sanshodhana Dikshit Award, Adarsh Pratibha Ratna Award, Best Principal Award, Young Scientist Award, young investigator scholarship award and so on. Now, I take this opportunity to invite our chief guest, Dr. K. Joshi Hanumantarkar, sir. Sir, now the podium is yours. It's over to you.
Thank you very much, Dr. Nagendra. Am I audible?
Yes, sir. Yes, sir. You're
Yeah.
clear,
So good morning to everyone. And first of all, thanks to our very good visionary in educationist, Dr. Niranjan Babu. Thank you for the kind invitation. And I am very excited to see, a very good speaker coming from Canada, Dr. Srinivas. Good morning to you, Dr. Srinivas.
Good morning, sir.
So there is nothing much to say because we have a wonderful speaker here who will be delivering, a excellent talk on drug discovery. But otherwise, everyone aware that pharmacy is one of, a very important profession which has been proving every time whenever there is a challenge across, the world, especially in the recent COVID. The pharmacy profession played a very, very significant role. Earlier, it was only prominent in drug discovery and process research, but now you see everywhere. Maybe it can be vaccine, it can be anything. So it's a wonderful area of research for everyone. And I am very sure, the audience of today's webinar will take a lot of benefit from the research and experience of Dr. Srinivas. Thanks to Dr. Niranjan once again, and thanks to all the friends over here. Have a nice time. Thank you so much.
Thank you.
Thank you, sir. Thanks a lot for your valuable speech. Now, I would like to invite my colleague, Redama Madam, to introduce and invite our today's speaker, Dr. Srinivas Kantam, sir. Now, Redama Madam, it's over to you.
Dr. Nagendra, your voice is not completely audible.
Yeah, I can barely hear your voice. Still.
Redema madam, I think your voice is not audible.
Can I audible now?
Yeah, there is no change.
Can you hear me, sir?
Yeah,
Oh, yeah.
now it is okay. Excellent.
Yeah. Good morning to respected principal, sir, chief guest, resource person, and my dear colleagues and delegates. Myself, Miss M. Redama, Assistant Professor, Department of Pharmaceutical Chemistry, Seven Hills College of Pharmacy. It's my pleasure to introduce today's resource person, Dr. Srinivas Kantam, sir, who is a medicinal and biomolecular chemist. Sir is a B Pharmacy graduate from Osmania University in the year of 2006. Later, Sir completed his MTech in Pharmaceutical Chemistry at VIT University in 2009. After his MTech, Sir worked as a project chemist at Indian Institute of Chemical Technology, Hyderabad. Later, Sir finished his PhD in medicinal chemistry from the University of Queensland, Australia in 2016. So after his PhD, Sir worked as industrial postdoctoral fellow in medicinal chemistry in partnership with Misentech at Simon Fraser University, Canada. Present, Sir is the senior scientist in medicinal chemistry at Misentech, Vancouver, Canada. Apart from these professional achievements, Sir also published many publications in the internationally peer-reviewed journals. Sir also achieved a valuable awards, which includes Endeavour Award, which is the Prime Minister's Australia Asian Income Postgraduate Scholarship. And Sir also received a MITEX, which is the Elevate Postdoctoral Fellowship in partnership with Misentech. So with this brief introduction, we are very glad to have you today, sir. Thank you so much, and we welcome you again, sir.
Thank you. Thanks for the warm welcome.
Thank you, Redama Madam, for introducing and inviting our today's resource person, Dr. Srinivas Kantam sir. Dr. Srinivas Kantam sir, now I'm handovering the session to you, sir. I'm making you as a host so that you can deliver and present the topic.
Okay, thank you, sir. Thank you so much. I'm gonna share my screen first.
Just a minute, sir. Just a minute.
Mhm.
Sir, now you can share the screen and start the webinar, sir, please.
Yep. Can you able to see my screen?
Yes, sir. Yes, sir. It's visible
Is-
clearly.
Is it full screen or just a presentation?
Not, sir. It's not full screen.
Okay, is it full screen?
Yeah. Yes, sir. Yes, sir. Now it's.
Okay. Is my voice audible?
Yes, sir. It's clearly audible.
Okay, that's great. Thank you so much. I'll leave it to me now.
Thank you, sir.
Thank you. Hello, everyone. Thanks for connecting me. It's such a great pleasure to connecting all you guys through my friend, Satish, who was my classmate at VIT University and asked me to present on this webinar. And I'm very glad. So I was accepted in my busy schedule. But after connecting, it's a pretty warm welcome to see all you big guys back in India. So let me introduce myself a little bit more in detail, which was not introduced by the respected chair people. I'm Srinivas Kantam. I'm basically from Warangal, old Andhra Pradesh, current Telangana. And I did my bachelor's in pharmacy from Polarity College of Pharmacy, Usmania University. I forgot to mention the college name, but I put the university name. Now currently working as a senior scientist at Masantech, which is a startup company. We are located in Simon Fraser University right now. And we are working on basic drug development. So our basic strategies is to develop the drugs, not to publish the papers. So we don't care about the publications, we care about the patents. And so far we have multiples of patents from our company side. Now I'm going to share with you guys our recent discovery of a bispecific drug conjugate technology for the treatment of osteoporosis. Just a minute. Close these notifications. I'm trying to get a pointer, okay? Wow. Can you see my pointer? Absolutely.
Yes, sir.
Okay. So in this today's session, I'm going to discuss about osteoporosis and the existing treatment options, followed by the discovery of bispecific drug conjugate technology. I hope this lecture will give you an idea how to start a drug discovery project from the scratch. And I'm not going into a molecular level or a cell level details. I'm going to give a very brief introduction, but I'll make sure that you can, think and you can write a proposal with your own knowledge by applying what are the principles that I'm going to say here. It might be useful in writing grants, writing publications, or, writing your own project. I think it will be useful. So these insights are very valuable, I thought. So let's go ahead. So a normal bone formation is equals to the bone resorption. Like every other cell, the bone cells also undergoes metabolism. That means the bone cells also gets older and they get replaced by the newer cells. So in a healthy human being, the bone formation is equals to the bone metabolism. The resorption, nothing but metabolism, okay? Whereas in osteoporosis, the bone formation is much lesser than the bone resorption. That means there's more metabolism than formation, which leads to a condition, a reduced bone density. So automatically, you will lose the bone minerals and decrease the density and mass of the bone. So once the minerals are gone, the mass is gone, the density is dropped down, then there is an increased risk of fractures. This basically happens at the age of 50 and above. Okay, so that's what the osteoporosis. So in this bone formation and bone resorption, basically there are two types of cells are involved. Osteoblasts, they form the bone, the osteoblast cells that are responsible for the bone formation, whereas osteoclasts, they are responsible for the bone metabolism. Okay, just keep remember. So in future, I'll talk about them. So now you can understand this disease, right? The bone formation is less than the bone metabolism. It's a very simple disease, very simple concept. So current treatment options. Let's see what are the treatment options available in the market. So the first treatment option, a parathyroid hormone analogs. So, okay, what they do, they increase the bone formation because in osteoporosis, there is a reduced bone formation. So parathyroid hormone analogs can help to increase the bone formation, which was decreased due to the disease. And the another class is bisphosphonates. What they do, they suppress the excessive resorption. They just block the metabolism of the bone cells. So these are the two different classes of the drugs are available to treat the osteoporosis. So, but why we are doing research? So what is the need of the drug discovery in osteoporosis if there are two classes of drugs are available? Okay, let's see. Parathyroid hormone. You know, we have the analog teriparatide, which is available in the market, but no any countries in the world, the therapeutic strategies are approved for this drug. That means it is not in use, but it was approved by the FDA, but it's not in use because of its toxicity. It is relatively expensive and a daily injection protocol. You have to inject into the bone. So you can't inject yourself like, you know, subcutaneous. You have to go to the hospital. You have to, you know, take admission and get the treatment. It's going to be super expensive. And recently it is identified that it's going to associate with the increase in the bone tumors. What it does basically, it increases the bone formation, right? At the same time, it excessively increases the bone formation, which causes a bone cancer. That's the major drawback for the parathyroid hormone analogs. So there is one vacant position in the drug discovery for this osteoporosis. So what is the second class? The second class is bisphosphonate analogs, the bisphosphonate analogs. The molecule which is in the market is alendronic acid, which is fine. So it's only 5 milligrams per day. You can take it oral. It is pretty good. It doesn't have any side effects. And its entire world is using right now all the osteoporosis patients. but it only stops the bone metabolism or bone resorption. It does not help in the formation of the bone that was lost due to the disease. Due to the parathyroid hormone problems, we're not using. So what we need now, so there is remains an important medical need for effective agents to induce healthy bone formation that might reverse the effects of the osteoporosis. So we can easily find out a drug discovery topic like this. Each disease has some problems. Each drug has some problems. Pick up the problem, and if you are able to fix it, then you are the hero in the drug discovery. So here we pick this problem of the parathyroid hormone, and we're gonna fix the problem, see how we're gonna fix the problem in the future. So we went back to the literature. Here we, in the sense, my supervisor, my previous colleagues, everybody who worked in this project, because this project was started 22 years ago. Like so many people worked in this project. I'm maybe like 7th or 8th person working on this project. Though initially this project was started with this first publication in 1985. The idea comes from this paper. This paper, the takeover message from this first publication is the prostaglandin E2 has been shown to stimulate bone growth in vivo in animals and humans. So apart from the parathyroid hormone, so we have a prostaglandin in the body. So it's naturally produces in the body. That's why we are not, we are all healthy human beings. We are not getting any osteoporosis, right? So who is controlling that? A natural hormone in our body, which is producing the bone. That's why we are surviving in a happy way. So the first publication gives a message that a prostaglandin E2, here is the chemical structure of the prostaglandin E2. So where it's going to act? So it exerts its effects through the EP4 receptors. But there are 4 different types of receptors that distributed through whole body. But we need a receptor that's specifically present on the bones only, because we don't want any other side effects. So you know that now prostaglandin E2 is going to increase the bone growth. Can you take a prostaglandin E2 directly? No, because it's going to act on different types of receptors. So we need to find out which receptor is present on the bone among these 4 subtypes. So if you look at the second publication, this work was actually guided by my supervisor, my current supervisor, and 22 years back when he was working at Merck. So they identified the prostaglandin receptor EP4. Okay, there are EP1, EP2, EP3, EP4 subtypes. Among these, the EP4 subtype. Okay, when this prostaglandin E2 attack on the EP4 receptors, the bone growth starts. The stem cells stimulates and produces the new bone naturally. So taking these 2 publications, trying to go to the next slide. So we know the key information. The EP4 agonist, prostaglandin E2, stimulate bone growth in vivo animals and humans. But Our problem is PGE2 is not selective for the PGE2 is not completely selective for the EP4. PGE2, it also affects on the PGE1, EP4, EP1, EP2, and EP3 receptors. So we got another problem now. So this problem is going to give us a project. That's our drug development, where we are going to start. So let's design and synthesize EP4 receptor agonists. So prostaglandin E2 is an EP4 receptor agonist, but it's not selective for the EP4 receptor. It also acts on the EP1, EP2, EP3. So we need to find out an analog of that one, which perfectly fits into EP4 receptor. Before I joined, like in this lab, before 2016, in our lab, we synthesized around 200 analogs of PGE2 as an EP4 receptor agonist. And they find out this mesenteric 1007, okay, as most potent and selective than PGE2 towards EP4 receptors. Okay, so can you use this one? Can you use this one as a drug? Probably as a yes, because it is more selective and it's more potent than the PGE2. But another problem is came in. Unfortunately, the MES1007 also associated with the other side effects such as gastrointestinal tract and hypotension. So which was actually lead to the complete abandon of this drug development process. So in 2016, what happened is, so our group was thinking on what to do. We have a better one than prostaglandin E2, but we can't use it as a drug because of two side effects. Okay, that means this drug doesn't have the specificity, doesn't have the selectivity. So we need to introduce a specificity and selectivity into this molecule. So what to do? Again, we go back to our equation of osteoporosis, which is bone formation is less than bone resorption. Now we have an alternate to the parathyroid hormone, which is MES1007, but it has its own problems. And what is the other drug in the market is bisphosphonate alendronic acid. It doesn't have any problems. Why it doesn't have any problems? Okay, we dig all the literature and find out this alendronic acid, it has a bisphosphonate group. That's why it's called as bisphosphonates class. The O minus, so in pH 7, like all these OHs becomes O minus or negative charge. All these negative charges with the 4 hydroxyl groups perfectly binds with the calcium ions on the bone. So whenever a person takes this alendronic acid tablets, okay, it goes into the blood, and within 30 minutes, it goes to the bone. After 30 minutes, if you take the blood and analyze for the alendronic acid, you'll see nothing. So it is highly specific for the bone. So we got an idea. Why can't we use this drug to direct this drug, our drug, the EP4 receptor agonist that we made in the lab, and target the bone? Okay, that's the idea generated. Okay, and we designed a new drug. We take our drug, we kept a linker and attached alendronic acid. Okay, what it does, if we give this entire a conjugate as a drug, it goes straight to the bone, and this alendronic acid part acts as a targeting agent, acts as a driver. It takes directly into the bone, okay? And then at the bone, the enzymes present surrounding the bone cleaves the linker and releases the drug moiety. And this drug moiety is a EP4 receptor agonist. It immediately goes and binds to EP4 receptor before it distributes into the blood circulation, because the blood perfusion rate at the bone is much lesser than other tissues. So there is no chance to escape from the active site. So finally, this is the structure. So we have our EP4 agonist. We have made lots of linkers. When I joined as a postdoc, industrial postdoc, I made like hundreds of linkers to test the stability of this conjugate, to test the release profile of this drug. You know, once the whole conjugate goes and attached to the bone, it has to release the drug. The esterases enzyme has to release the drug. So what is the rate of the release? How long it takes to release? When we talk to the doctors and the key opinion leaders we usually attend key opinion leaders meetings in the hospitals. Here in Canada, the most of the hospitals, they will arrange key opinion leaders meetings. If you can register there, you can go there and doctors will tell their problems. And you can share your problems too. It's free of cost. Government does that one. So when we talk to the doctors, they said that they want a half-life of 7 days. The drug should release in 7 days. 50% of the drug should release in 7 days. So to get that one, to get 50% of the release in 7 days, we changed lots of linkers. I finally fixed this linker, and it releases the T half-life is for this process is 7 days. We have all profiles, like we have conjugates from like 10 minutes to like 25 days. We made all those things, and we picked one analog, that is this drug, this conjugate. So with the help of alendronic acid, that's our targeting agent. We have a linker, we have a drug moiety. Okay, we are giving this one in IV route for the preclinical studies, but with the clinic, we already applied for the phase 1 trials with the FDA, and we applied for the subcutaneous, not the IV, because IV needs, again, you need to go to hospital, or at least you need to see RMP doctor to get a injection, whereas subcutaneous, you can do it yourself. So this drug, finally, this conjugate gives you a drug with the help of esterase and the alendronic acid generated EP4 agonist directly binds under the EP4 receptor. Okay, I will tell you how it generates the bone, okay, in later slides. So this is the basic concept. So what we got here is a tissue specific, very selective to the bone. And also it is a receptor specificity. It's not gonna attach to any other EP4 subtypes, because on the bone there is only one subtype, that is EP4. So here you will see the same concept, but this is our proprietary material we have put in our patent. In patent, we write a bit different than what we write in our regular notebooks. We show this drug conjugate as a instead of alendronic, because alendronic acid itself is a drug. So you can't claim as it is your drug. So then patent right problems will come. So we have we took the alendronic acid and the linker part together and called it as a targeting moiety. So we need to learn all these tips for the patent purposes, because otherwise you need to face many court cases. So we call it as a targeting agent, and whatever the, you have the the carbonyl group, the conjugate bond, and that's the linkage, and we have a drug moiety. We did not change anything, but we presented it differently. And then how it works. So this is a bond. You give this conjugate, inject through the IV route, it goes to the blood and immediately disappears in the blood. Where it goes straight to the bone, because the targeting moiety directs the drug to the bone. And then, okay, this conjugate bond is going to cleave with a half-life of 7 days with the help of the enzyme esterases. And once the drug is released, it goes and attached to the EP4 receptors present in the stem cells. And these stem cells releases the osteoblasts. You know the osteoblasts, right? I told you in the first slide, the osteoblasts are responsible for the bone formation. Once the osteoblast is formed, this process is completely natural because we are just stimulating the stem cells. The stem cells releases the osteoblasts that are natural, not synthetic. Parathyroid hormone was the overgrowth. It keep on stimulate until it generated tumor of bone cell, which is osteosarcoma. We did not see that one with our drug because it directly activates the stem cells. Then stem cells takes the natural path and gives us osteoblasts. So you can question the release profile. So here is the conjugate. So what are the, cell bonds here? There's one ester bond here, another ester here. Here you have a carbamate. Okay. So the ester bonds are more susceptible than the carbamate bonds. So that's why this drug first cleaves at the ester position. So you can ask me which ester cleaves first. Yes, both cleaves instantly. I mean, in 7 days. Because of this allylic one, it's not allylic, the alkyl carboxylic esters cleaves much faster than the allylic OH one, the allylic alcohol one, because these allylic alcohol lone pair electrons are in conjugation with the double bond. That's why this guy cleaves second, this guy goes first, this guy goes second, this guy goes third. So what are the rates? Okay, the first rate in 7 days, we give you the drug, okay, and give the rest of the piece. What does the piece does? Then it undergoes the cleavage at carbamate in 28 days, gives alendronic acid and the remaining linker. So by the 28 days, usually this drug displaces from the active site and it eliminates from the body. Whereas in the other pathway, we studied this one, and it takes 22 days to get some part of this molecule as well with the cleavage of carbamate. So in majority, we are getting our drug that is 2A, that's the prostaglandin analog of what we described before. So we're delivering that one in 7 days. So how to measure these things? Like, we give this drug in like 5 milligrams per kg weight. When it distributes in the body into different parts, at bone, we have like a nanomolar concentration, which technique we'll use to determine the concentration of the drug. You cannot use HPLC. You can't use the gas chromatography. You can't use the mass spec. You can use mass spec, but you can't quantify at that level. So what we did is we introduced radioisotopes. And where my specialization comes in, I'm very specialized in synthesizing radioisotopes and radiopharmaceuticals. So what I did is I introduced a tritium instead of hydrogen and the aromatic ring, and also a C14 isotope on the linker. Okay, in order to trace these like pieces or the drug or all the fragments. So we gave whole conjugate. We put these two isotopes into the molecule, and then we inject the molecule to the rat or mice, and then we take the blood and see different, like we take the blood in different hours, and we measure the radioactivity in the blood. And at the end, we sacrifice the animal and we take all parts of the animal and then isolate the radioactivity into a solution. Okay, then we measure the radioactivity in that solution. So we collect each and every part of the body, like bones, all tissues, okay, all organs. We separate them individually, and then we isolate the radioactivity, and we can measure that. Even if you have 10 to the power of minus 10 picomolar concentration, you can measure the radioactivity. You can't miss it. So we introduced C14 and tritium into the molecule. And if this bond cleaves, if the ester bond cleaves instantly, once you inject it, that means the actual drug moiety did not travel to the bone. It was in the blood. If that happens, you should see the radioactivity, the tritium radioactivity in the blood. But we never saw that one. When we sacrifice the animal, we take the bones and isolate the tritium, we see the tritium. We never see the tritium in the blood. And also, just to look for the fragments, we also studied this C14. And we also found the C14 on the bone as well. So that's why we use a double labeled conjugate to measure the distribution study, usually like, your pharmacokinetics. For this, especially for the osteoporosis, pharmacokinetics is quite different from the regular pharmacokinetics. You need to sacrifice the animal, you need to take each and every part and then analyze each and every part. Then only you will get the FDA or any other approvals. If you do the regular kinetics, pharmacokinetics, you won't get the approvals. So drug release profiles. I'm just going to show you, but I'm not going to detail. I'm just give you like overview here. And the y-axis, the percentage of the original tritium dose, that's the drug. So this is the blood samples. We have given the radioisotope drug to the animal, and we took the blood in different time intervals, like 2 hours, 4 hours, 6 hours, and 8 hours. Okay, you see, by 2 hours, came to 0 in the blood. There's nothing in the blood. Oh, where it went? It should be in the bone. So once we sacrifice the animal, Okay. We studied different organs. And we separated especially bones versus liver and other tissues. Because, why? Because liver, whatever you give it to the blood, you give IV or whatever the route, it goes to the liver faster than the bone. So it has to go through the liver. That's the first principle. I think every pharmacist know about that one. So that's why you see lots of drug molecules, lots of tritium in the blood, in the liver. And as the bones, the second majority is in the bones. And the third one is others. The others includes like all other tissues, including the feces and urine. We collect feces, we collect urine and everything. We don't leave anything for the animal. And the original dose versus days post date. We want to see how long this tritium or how long this C14 isotope, the drug is going to stay in our body. It stays up to 30 days in our body. Okay. The half-life is 7 days. Okay. But it shows up a little fragments stuck to the bone. It's there like around 30 days. You see the minimum activity at the 30 day, 28th day. Here I gave you some information about the C14 as well, because I told you the C14 labeling before. Just to get an idea whether this drug or this conjugate is cleaved in the blood before reaching the target, that's a bone. So, the radial, as soon as you injected the y-axis, you see the percentage of the radiolabile dose in the blood versus time. It instantly decreases the concentration in the blood. It travels to the bone. And also the radiolabile dose in bones. The C14 is increased, whereas the tritium is decreasing slowly. That means at the bone, the conjugate reached the bone, and it's slowly cleaving and giving a drug, and it's reacting on the EP4 receptor and generating our new bone. So, and the third one, the percentage of the radiolabile dose in the liver. So as I shown you in the previous slide, there's large excess of drug in the liver. You may ask me, hey, can you get a liver toxicity with this drug? No, because slowly it eliminates from the liver. You can ask me a question, how long it takes? Usually, most of the drugs takes, like even if you take a paracetamol, it stays in the body, in the liver for 50 days. You don't imagine, but it takes 50 days to paracetamol excrete completely from the liver. So this drug is like in the optimum level. It's within the levels of WHO and international guidelines. How safe is it? Is our drug safe? Yes. It avoids systemic exposure and side effects because directly targeting the bone. And we have, apart from the mice and rats, we have also completed the GLP toxicity studies in NHPs. NHPs doesn't meaning that the bigger animals, like we tested in monkeys, chimpanzees, and we did not find any unexpected toxicities. So finally, we recently, not recently, 2 years ago, 2021, we have published all this work. We have patented this one. We published this work in the ACS Pharmacology and Translational Science. There's a publication here. I can give you the slides if you want. I can share the publication. You can go through it. And you can read the process of drug development. And here are the corresponding author. You can see this guy is my boss, Robert M. I have been with him from last 7 years, 6 years, 6.5 years. He's the director and founder of the Mesentic, and he was my postdoctoral supervisor as well. And yeah, and the other people worked on this project were like Marian, and she was the first synthesized the non-radioisotope version. And the Gong Chen is the director of our medicinal chemistry, and I'm Srinivas Kantham. I was involved in the radioisotope drug synthesis as well as analyzing the radioisotope materials. I can handle both chemical and animal samples. And the Sun and Gloner, they are the dentists. So they are doing the bone sections. They work on like bone dissection, and they get beautiful pictures for our FDA approvals. And he is my boss. He's in the background of all this work, as the entire idea belongs to him. And he was the former Merck chairman of MedFirst Canada. He was retired and is back to the research into university. And so far he established like 6 or 7 companies. Once you get FDA formation, like you will push the molecule into phase 1, phase 2, and you will sell it for $1 billion or $1 million, $100 million, you know. So most recently, we have also used this similar kind of technology, but we did not use the alendronic acid. But the concept is very similar. I haven't disclosed this structure yet. We haven't disclosed anywhere, but we just published our biology data in Nature Cell Biology, and it was appreciated by many great scientists around, because for the inflammatory bowel disease, there is no proper treatment. Actually, there is no proper diagnosis for the inflammatory bowel disease. We are going to come up with a proper diagnosis for the inflammatory bowel disease as well as treatment. So we are in the process. We are in the preclinical stage at this. We are going to take another 2-3 years to get this molecule out. Maybe I'll present in few years later regarding that molecule. So this technology is bispecific technology. What I presented now is quite novel. It resembles like a prodrug, actually. I think you know what prodrugs. It's an inactive molecule which goes into the body and gives an active molecule. But that active molecule flies everywhere. Okay, with this bispecific conjugates, okay, you target the site and then release in that area before it distributes into the other compartments. You can get a better idea on drug design, designing a new drugs. Even for the older drugs, you can repurpose the drugs. You can also change the formulation of the drugs. Like if you have a tablet form, you can make it into subcutaneous, even though it is not water soluble. Okay, because you can introduce soluble groups later. So that's how a bispecific conjugate. Nowadays, lots of companies are following this one. There are a lot of patents going around. Lots of FDA approvals in phase 1, phase 2. There are like more than 100 molecules of bispecific conjugates in phase 1 study right now with US FDA. So this is a completely new trend to direct the drugs, either new or older drugs. I think this will be helpful, in thinking of new projects. Yeah, that's all for now. Thank you so much. Thanks for having me, and I'm happy to take any questions you have. Thank you.
Thank you, Dr. Srinivas, sir.
Welcome, sir.
So now I will call upon for our delegates for any questions or queries regarding the session. Dear delegates, if any one of you having any queries regarding the topic, kindly ask for the queries. Or you can send queries in the chat box also.
Yeah, I can open that one, and yeah, I can look for questions.
Okay, as we have not seen any kind of queries or questions either in the meeting or in the chat box. Now let's move further. Thank you very much, Dr. Srinivas Kantam, sir, for your valuable inputs regarding the cutting edge strategies in drug discovery, especially on the topic of osteoporosis and how to handle it and the discovery process. And even importance of going for patents instead of simply publications. You had narrated things very well, sir. And now I'm going to hand over the vote of thanks session to Dr. Satish Kumar, sir. So please hand over the session.
A very good afternoon to Nando. Myself, Dr. Satish Kumarji, working as a professor in Seven Hills College of Pharmacy. So it's my immense pleasure to deliver vote of thanks on this occasion of webinar. I sincerely thank our today's chief guest, Dr. Joshi sir, Principal, Sharada Blask College of Pharmacy, Mysore, Karnataka, India, for his presence and valuable words to every one of us. Next, I want to thank wholeheartedly to our today's speaker of the session, Dr. Srinivas Kantam sir, Senior Scientist, Mesentech, Canada, for his wonderful and an informative session. And it is an honor to have the opportunity to express thanks to all the participants and extend my sincere thanks to our beloved principal sir, Dr. M. Niranjan Babu sir, and correspondent ma'am, Mrs. Somlata ma'am, for giving this opportunity to organize today's international webinar on cutting edge strategies in drug discovery. Thank you one and all again for joining with us. And we have shared the feedback link in the chat box. So kindly fill up the feedback link and we will share it to your corresponding email IDs also. And you will receive your certificates from Tuesday onwards. Thank you one and all for joining with us.
Thank you, Sadish.
Thank you, sir.
I thank one and all who took their valuable time to attend for this international webinar on cutting edge strategies in drug discovery. And I thank heartfully our chief guest, Dr. K. Joshi Hanmantachar, Principal, Sharda Vilas College of Pharmacy, Mysuru, Karnataka, and our today's resource person who had given his very valuable inputs and knowledge, Dr. Srinivas Kantam, sir, senior scientist, Mesentech, Vancouver, Canada. And with this, I'm going to close this webinar session with heartfelt thanks, even with the support of our principal, Dr. M. Niranjan Babu sir, and correspondent madam, Mrs. M. Sumalata. Thank you very much, one and all. And soon we will meet again with another new topic in coming days. Thank you very much. Thank you all.